Okay, so my aims for the next half an hour is we are going to look at what are psychedelics. We're going to look at some of the common effects, occurrences, and their pharmacology. We're going to look at psychedelics and neurons. We're going to look at how neurons function, psychedelic 5-HT2 antagonism, affinity, and functional effects. And I'm going to have a look at psychedelics and networks. Particularly, I'm going to cover the default mode network, visual perception, ego dissolution, and connectivity. So, without further ado, I'm a big fan of etymology, so I wanted to start by having a look at where the word psychedelic comes from. So, the word psychedelic was first coined by English psychiatrist Humphrey Osmond, who is pictured over here, in a letter to Aldous Huxley. Osmond had been experimenting with LSD and mescaline and had noted some similar effects of schizophrenia, mainly that and distortions of perception. And psychedelic comes from two words in ancient Greek. Psyche, from the ancient Greek psyche, for mind and soul, and delit, from delos, for manifesting, and thus psychedelics as soul-manifesting experiences. So, what are classic psychedelics? These include stuff like LSD, DMT, and psilocybin, and they have some common characteristics. They are 5-HT2A agonists. 5-HT2A, as we'll cover soon, is a type of serotonin receptor. And here is a little photo. You can see that little bit blue there. That is LSD inside of the 5-HT2A receptor. They cause perceptual distortions and hallucinations, as well as the possibility for religious or mystical type experiences. And there are all sorts of these in nature. I thought I'd highlight a couple of them here. So psilocybin cubensis, these are the most common mushrooms, or psilocybin-containing mushrooms. Psychotria vividris, this grows in South America and contains DMT. You've got the Colorado river toad, Bufo alvarez, which produces 5-MeO-DMT. This guy down here, this is Hawaiian baby wood rose, which produces LSA, and mescaline-containing cacti over here, which grow in Central America. So, before I start going into how psychedelics work, I wanted to do a little bit of a background on the brain, and we're going to start right at the top, and we're going to move right down to neurons. So, the brain is a big 3D computer with all sorts of different regions that are involved in all sorts of different tasks and communicate with all sorts of other regions. And there are plenty of ways that we can break the brain up. We can break it down into 4 basic lobes: the frontal lobe at the front here, your temporal lobe on the side, your parietal lobe at the top, and your occipital lobe at the back. We can separate them based on different sulci and gyri. The sulci are these little indents, or the ridges, and the gyri are the bits up top. Or we can break it down based on their function. So if we have a look at the top of the brain over here, we've got the motor area. At the back of the brain up here is where we might be perceiving vision. And in simple, the brain is a big 3D computer with a complicated but highly ordered architecture. Now, if we have a little bit of a zoom in, we get 2 cortical columns. And cortical columns are basically the axes in which problems are solved along. So, if we have a little zoom in to the brain over here, hopefully you can see that in blue is this kind of very outer layer, or layer 1. And as we move in, you can see layer 2, layer 3, all the way up to layer 6 in pink there. And each of these layers does a little different thing, and you can see they contain different types of cells. And these are the basic building blocks which allow for computations to occur within the brain. Yes, the cortical columns are these mini circuits which allow for the computation. And if we zoom in on these even further, we can see that in each layer inside of these, there are all sorts of neurons of all different shapes and sizes. And so, information typically starts at level 1, and it's generally transferred down over here. And when you get down to the bottom bit here, these neurons are kind of collecting information from all of these neurons up here and compiling it all along. And yeah, you can see that all of these different ones contain different types of functions. You don't need to learn or anything. But basically, cortical columns are made up of different layers, which have different cell morphologies and do different sorts of functions. And we will be discussing these a little bit later when we get on to psychedelics. And if we zoom in on one of these neurons in particular, here is a pretty classic example of a neuron. you can see it's got a cell body over here with its nucleus. It's got dendrites here. Now these connect onto other neurons down here and receive information. So information comes in from other neurons there, and then it sends along information across this thing called an axon, which connects to other neurons. So over here, this is another neuron, and this is how the brain basically communicates, through electrical impulses. Now, so neurons are the basic building blocks of the brain. We have a huge number of them, about 200 billion in the brain, which is almost impossible to fathom. And on average, each of these is connected to about 7,000 other cells. So there's a huge amount of neurons and connectivity between them inside the brain. And if we have a little zoom in over there where it says the synaptic terminals, we can have a look here at something called the synapse. So, up the top here, electrical impulse comes in from the first neuron. That causes these neurotransmitters to be released, and they come across here. And they bind to the second cell over here, to these little receptors. And that propagates the signal or transfers it along. And then electrical activity triggers in this neuron, and then presumably they'd synapse onto another neuron, and then you get this progressive transfer. And if we have a little zoom in on these receptors, they look a little bit like this. Now I've picked out the 5-HT, or the serotonin receptor in particular, because it's relevant to psychedelics, but hopefully what you can see is that when there's no 5-HT bound, this channel is a little bit closed, and then the second these receptors bind, this little thing opens, allows some ions to come in, and these ions are what kind of reignite the signal in the next neuron. So, synapses are tiny junctions between cells, or between neurons, where neurotransmitters can relay electrical activity. And this electrical activity is how the brain is communicating. So, if we have a little zoom in even further, we can have a look at serotonin receptors. Again, there's a lot of information, you don't need to worry about it. The point of this is serotonin receptors come in all shapes and sizes, and that one molecule of serotonin can bind to all sorts of these different types of receptors, so the 5-HT to 1A, 1B, and so on. And hopefully you can see here they are found in different parts of the brain, and they have all sorts of different effects. So there are lots of different types of receptors which serotonin can bind to, and they're involved in all sorts of different processes. Now, I mentioned a little bit earlier that the 5-HT2A is the one that we're particularly interested when it comes to psychedelics. So we can have a little look here, and we can see that it's involved in cellular excitation and muscle contraction, which isn't too important, but the cellular excitation is totally important for psychedelics. So, here is a photo of serotonin, 5-hydroxytryptamine, or 5-HT for short, and here are some photos of some psychedelics. You've got DMT over here, psilocybin and LSD. And hopefully what you can see in all of these is that they have this really similar backbone. They contain this type of thing across all of them. And this is an early hint that psychedelics are presumably going to be acting through some sort of serotonergic mechanism, because they do look very similar. And classic psychedelics do in fact mimic serotonin within the brain. And here's a nice photo. This here is a reconstruction or a visualization of the 5-HT2A receptor. And if you see over here, that's psilocybin kind of tightly bound within there, looking very pretty. So, if we have a look at some graphs, 5-HT affinity and potency. What this graph is showing here, each of these little dots is a different drug. This comes from Shulgin in around 1950s and then reprinted by Clayden in 1984. And what you can see is as the affinity, or basically how well they bind to these serotonin receptors, goes up, the potency also goes up. So at the top over here we've got LSD. Now some of you may know LSD is particularly potent. You need micrograms of it to have a really, really vivid visual experience. And then down here at the bottom we've got mescaline. Mescaline can still give you a really good time, but you need a lot more of it to feel the same effects. And then this is a really cool study done a couple of years ago. Each one of these dots is an individual participant on the study taking a psychedelic. I believe it was psilocybin. And what we are looking at is occupancy of these 5-HT2A receptors, or basically how many of their receptors are occupied with psychedelics. And what you can see is as this occupancy increases per participant, they have a more intense trip. Or if you have a higher affinity, you get more potent effects. And this is good proof for us to suggest that this is indeed working through the 5-HT2A agonist. just as quickly, if you've got any questions, there will be a Q&A at the end, but if you want, you can also stick your hand up at any point. So, 5-HT2A in the brain. This receptor is not found everywhere across the brain, and in fact is distributed in specific regions. What you can see here, this is a PET study, PET, and this is showing the density of 5-HT2A across different parts of the brain. The areas in red are places where there's a lot of 5-HT2A, and the areas in blue are where there's not a lot of it. Now, hopefully you can see there are a couple areas in particular. This front of the brain here, the frontal lobe, or the prefrontal cortex, and also this cingulate cortex, which are really quite heavily involved. So, or very dense in these receptors. So we might imagine that tasks that involve these regions would be altered on a psychedelic experience. And just as a nice little one here, if we have a look up here, this is your motor strip over here, nice and green, and that's the reason why psychedelics don't mess with your movement and you can still walk on them and stuff. So the highest density is in the neocortex, or the area on the outside of the brain, and specifically the prefrontal cortex and the cingulate cortex. And these two areas are really, really important for all sorts of executive control functions, and the prefrontal cortex in particular is the master controller of the brain and makes sure that all the other regions are working together. Yes?
Can I ask, I'm interested that the visual cortex, I'm assuming, is not as highlighted as I would expect it to be. Can you speak on that?
Yeah, so that is interesting. We will get onto it a little bit. Psychedelics do indeed cause increased activity in the visual cortex, at least to some extent. If you look right here, that is still, I believe, that's around V1 of the primary visual cortex. So there is a little bit, but you're absolutely right, it's perhaps not as dense there as you might initially expect. And we'll get onto that. And the main reason for, you might be wondering, well, why do they cause these hallucinations then? The hallucinations are presumably to do with the prefrontal cortex up here, as we'll see in a little bit. When sensory information comes in, or vision is processed here, but it moves up the brain towards the front for kind of checking and quality control, and that quality control is kind of what's thrown off in a psychedelic experience. Is that alright? Lovely. So, if we have a look, particularly, you might remember that we said that there are these cortical columns, and there's all sorts of different cell types, and the cell and the layers do different types of functions. So, here's a lovely figure from Weber and Andrade in 2010. What you can see is these are the upper layers, so layers 2 and 3, and as we move down, we've got layer 4 and layer 5A and 5B. In red, we have stained the 5-HT2A receptors. And hopefully what you can see is there's not a lot of them up here, but there's a lot of them down here. And if we have a look and remind ourselves over here, these are these kind of pyramidal neurons, which you might have heard, and these are where the 5-HT2 receptors are on. It might be sitting on these little orange ones over here. Again, as a reminder, these are presumably going to be in the front of the brain here. Now, these layer 5 neurons are super duper important, and they integrate all the information that's being processed up here, and they combine information across cortical columns, and they're also the regions that then send off the information from one part of the brain to the other part of the brain. So these are really important for integrating and associating all sorts of information. So we might imagine that when psychedelics come along and bind to these and cause this disruption and wave of intensity, that this association and integration stuff seems to go wrong, and it absolutely does. So, psychedelics interfere with intercortical communication. That's communication between two separate regions of the brain, particularly in integration and association networks. So when I'm talking about integrating and association, I'm thinking about combining multiple different visual features or stimuli, combining multiple different types of sensory modalities, and all sorts of this stuff. Going to move on to some brain regions now, move away from our neurons and onto regions. Now, out of curiosity, do you guys, raise your hands, do you guys think psychedelics increase neural activity or decrease? Can I get a hands up for increase, please? Yeah, so most of you. Actually, it's not the case, and psychedelics make the entire brain significantly quieter. And you can see here, this is an MRI photo, which records bulb signals, and you can see that this is going down, which is really surprising. This is the first MRI study on psychedelics, was in 2012, and the authors were really surprised to see that the brain was actually a lot more quiet. And when you think about psychedelics and these really intense, powerful experiences, that was really surprising for them, and they had to try and reconcile that. And also, it's not everywhere that this quieting happens. In fact, it's in really specific regions. In particular, at the front of the back of the brain here, we've got the ventral posterior cingulate cortex. We have the anterior cingulate cortex, or the medial prefrontal cortex at the front here. Putamen, and the MPFC over there again. So, psilocybin in particular produces region-specific decreases in activity. And if we zoom in on one of these areas in particular, this ACC over here, what we can see is actually the more decrease you get in this area, the more intense your effects are. Well, basically, this axis over here is showing you the decrease or the change in cerebral blood flow. And as this is more of a decrease, the intensity increases. So each of these dots is a participant, and the ones that have this biggest drop get the most intense effects. So a decrease in this anterior cingulate cortex blood flow correlates with the intensity of effects. And again, this is more proof to us to suggest that this is actually the way that psychedelics are working. This isn't just some weird artifact. And moving away from regions, because networks in the regions of the brain are not individual and they are constantly communicating with other regions of the brain. And one network you might have heard of a lot, if you are kept up to date with the psychedelic scene, is the default mode network. Now, the default mode network is also known as the task negative network, and it's this network that is involved, or regions of the brain that are kind of active when you're not doing anything task oriented. So if you're on a walk home, just kind of having a little think in your head and you find yourself mind wandering, that's the default mode network. What I'm doing right now, trying to talk, my default mode network would be very quiet. And there are a couple important regions of this: the dorsomedial prefrontal cortex up here, the posterior cingulate cortex we've seen again, and the angular gyrus over here. Now, what is really cool is ayahuasca causes decreases specifically in these areas. So you can see over here, this is your dorsomedial prefrontal cortex, and this is your posterior cingulate cortex. So, psychedelics are decreasing the activity within this default mode network region. So let's have a think. Let's have a look at what this default mode network is involved in. So, first things first, the DMN and ego dissolution. The DMN is thought to be related to your sense of self, and in particular, the connectivity or the communication between the medial prefrontal cortex, or the red dot over here, and this blue dot here, or the posterior cingulate cortex, is thought to mediate sense of self. Now, that might be a little bit weird. Quite frankly, sense of self in general is bizarre, but it's thought, at least, that communication between these two regions is what gives us our sense of self. And what's really cool is that, there's a study over here, this is people taking psilocybin, and the bigger the decrease in the connectivity between these areas, the more dissolution you get, or ego dissolution. For reference, if someone is not familiar with ego dissolution, this is the loss of sense of self. And it seems to be that when these two regions stop communicating, that's when you get this ego dissolution. So here's a nice little mechanism for how that could possibly be working. And yeah, psychedelic-induced ego dissolution is related to this reduced MPFC-PCC connectivity. Now, the default mode network is plenty more important than just being this kind of mind-wandering or task-negative network, and regions of this are known to be overactive in depression. Now, psychedelics and depression are talked about an awful lot, and we shall come on to that a little bit more. But here is a photo showing you, this is the average of a bunch of patients with depression, and you can see that this medial prefrontal cortex is really, really overactive. So we might think, and remember that psychedelics are decreasing activity in this network. Well, if we're decreasing activity in this network, maybe that's going to be related to depression. And this absolutely is the case. This is a really cool figure from Berman. And what this shows is that as the connectivity between the two areas of the default mode network, the SCC, which is also known as the ACC, or the medial prefrontal cortex, and the PCC, as this decreases, their ruminations decrease, which is really cool. So reduced activity in the prefrontal and cingulate cortex may be associated with this reduction in depression symptoms. And again, I'm sure many of you would have heard about psychedelics being able to cure depression and all sorts of these things. But here is a plausible mechanism for how this could be working. And just to show you that I'm not alone, this is a search I did a couple of days ago. People are very excited about psychedelics and depression. So this is a Google Scholar, and there's 31,000 papers on this, or articles that mention this already. So, wanted to have a look now at visual perception and hallucinations. And in order to understand how hallucinations happen, we need to understand how perception works. So perception is a process of probabilistic inference, where you are taking in incoming sensory data, so visual stimuli, like all of you, and combining this with prior expectation. Now, if anyone is brave enough, what can anyone see in this photo over here? Does anyone see, does it look 2D or 3D? Can anyone see anything 3D in any of these? Yeah, does anyone want to tell me why they think that some of these look like they're sticking out and some of these look like they're sticking in? Because of the shading. Any idea why? So, if we have a look at the ones that are all sticking out, what do they have in common? Absolutely, right? So we are subconsciously assuming that light comes from above. So when we see a shadow at the top here, we assume it's being curved in. And when we see a shadow at the bottom, we're assuming it's bulging out. So this is a nice little bit of proof that perception is a process of probabilistic inference. It's more than just the sensory information, and it's all about combining this with your prior information and expectations. Now, perception is commonly thought of as a hierarchical process. This means that it moves from region to region to region to region, and as we move up, they get increasingly more complicated about what they are representing. So, if we start here, light photons will hit your eyes in the retina over here. They move to the LGN, or the lacular geniculate nucleus, move into the back of the brain, V1 and V2, or your primary and secondary visual cortices, and they move up the dorsal stream and down the ventral stream to the front of the brain, where some sort of choice is made typically about what we're seeing. So visual perception is hierarchical, and at each layer the representations of stimulus are increasingly complex. So in areas like V1 and V2, these might be looking for really basic things, so borders, lines and colours. Whereas when we get up to V3, or further up here into the parietal lobe, we might be looking at general different things, so maybe a watch as opposed to just the bare part of that watch. Now, one very popular theory related to perception is called predictive coding. And what predictive coding suggests is each layer of this hierarchy holds a prediction about what you are going to expect to see. So before I even look over here, I have some subconscious expectation of what I'm going to see. And the idea that predictive coding suggests is that each layer of this hierarchy holds this prediction. And what happens is sensory input comes in down to the bottom. It's compared with the prediction over there, and it's transfer, or basically, you look at what you see versus what is predicted to be seen, and you transfer this thing called a prediction error, or the difference between those, to the next layer. And then the same thing happens where you compare this prediction to this prediction error, all the way up until the difference is reconciled. And so when stimulus are presented, each layer compares their prediction to the incoming sensory inputs. And this is what predictive coding suggests, and this suggests how perception occurs. Now, what you might see is there's also some arrows going down. And what we believe is that areas higher up in the hierarchy are sending predictions down to these lower layers about what they can see, based off of past experience and expectation and all sorts of stuff like that. Now again, we're getting a little bit far ahead of ourselves, but if we remember that psychedelics are causing this decrease in activity in the prefrontal cortex, well, we might start to think that these predictions, these downward predictions, are going to go a bit haywire. And they exactly do that. And this brings us to one of the biggest models in psychedelics. It's called reboots, or the relaxation of beliefs under psychedelics. It's not reboot, it's the S from psychedelics for some reason. And what this suggests is that these predictions are shut up by a psychedelic. So over here at the top, you might imagine sensory input comes in, and we get these bottom-up prediction errors, and at the same time as they're moving up the hierarchy, we are getting top-down predictions. Now, under psychedelics, these top-down predictions kind of stop. So we have this unguided process of just sensory input being accumulated and going up the hierarchy with no control center. And again, we can consider hallucinations as kind of a mistake where the brain is going, well, that actually shouldn't be there and shouldn't be looking like that. but there's nothing to tell the brain that because of these top-down predictions being silenced. And so psychedelics relax these top-down predictions or beliefs, which may result in uncontrolled perceptions and hallucinations. And furthermore, getting back to your point here, psychedelics actually increase the activity in this primary visual cortex. Again, this area that we think looks for borders and shapes and basic stuff like that. What you can see is, as the activity in this area increases, each of these red dots is a participant, and the more activity they get, the more complex images they report under psilocybin. And as I said, V1 looks for stuff like borders and contrast. So these types of geometric illusions, which are quite common in a psychedelic experience, could very possibly be explained by this increase in V1 activity. And again, as a reminder, this is alongside this decrease in activity in these areas that are associating and integrating in control. So this increased activity in the primary visual cortex, alongside the decreased activity in these association hubs, may cause these complex hallucinations that are the staple piece of a psychedelic experience. And these, and psychedelics relax beliefs more than just in basic perception. And again, in emotional perception, so how we perceive emotional stimuli and stuff like that, these may also be relaxed. And in particular, cognitive rigidity, or these kind of fixed into one mindset, is very well associated with poor mental health. And to give you some examples of what this might be, in a patient with treatment-resistant depression, they may have this really, really strong prior expectation that the day is going to be rubbish. Similarly, in a patient with a chronic pain disorder, they would have this expectation that that part of their body is going to hurt. So rebus might come along here, and rebus could possibly be relaxing these beliefs. And again, we do think this is the case. And psychedelics can relax beliefs beyond basic perception, and can allow for the resetting of these rigid priors in mental health disorders. And so psychedelics may be the key to unlock this cage, and to open your mind is a very common phrase within the psychedelic scene. Almost done, got a couple more slides. The next one is looking at global connectivity. Now this is a very famous and very beautiful photo. Each of these little circles represents a different region of the brain. And there's two of these here. You can see the placebo, or this is what your normal brain looks like. You can see that there are certainly some regions that communicate with other regions, but in general, there's not a lot, and there's absolutely regions that only communicate with some parts and not the other parts. And then you look at psilocybin, and every region is communicating with every other region, and there's all sorts of going on. And so whilst psychedelics decrease the overall activity, they increase global connectivity, especially between regions that don't normally communicate with each other. And this is why we think we get some weird effects like synesthesia. So synesthesia is where you have sensory modalities that are kind of integrated. So you might be able to taste colours or smell touch. And these are known to occur under psychedelic experiences. So this is a possible explanation of how this might occur. Another big word that you hear all the time within the psychedelic scene is neuroplasticity. And psychedelics, or psychoplastogens as they're labelled in this lovely figure, are known to increase synaptogenesis, or the formation of new synapses, neurogenesis, the formation of new neurons. And you can see here that under psychedelics we start with this neuron here, and then it's making all of these new spines or synapses and all sorts. And very recently, because I did want to feature this, it was recently shown that this happens through an intracellular 5-HT2A mechanism. Up here you have a cell that's been given DMT and one that's been given psilocybin, and you can see they've got all sorts of these branches. Both of these are membrane permeable, or they can go inside of the cell. Now when you take derivatives that can't go inside of the cell, you can see that these don't really seem to be forming all of these new branches. So psychedelics induce neurogenesis and neuroplasticity, and these are essential for cognitive flexibility, so this idea that we can move from one belief to the other, as well as in general learn. And the final way that we might be able to think about this is in a little bit more of a chemical way, thinking about energy landscapes. Now this might look a little bit complicated. What I want you to imagine is that both of these little troughs here are two different beliefs or perspectives or outlooks on life. And if we start here, under the normal brain, you can see that it might be quite difficult to get out of there, move over this little ridge, and then move into this new mindset. We can say this has a high transition energy. Now, what was shown very recently by Singleton in 2022 was that psychedelics lower this energy landscape. And in practical terms, we might think that that means it's easier to move from this to this. So, psychedelics lower the energy needed to move from one belief to another. Again, this might be how psychedelics are allowing people to change their mind. So, to conclude, psychedelics bind to layer 5 pyramidal neurons, the 5-HT2A receptors in these, specifically in the prefrontal cortex and the posterior cingulate cortex. Now, as we said, these are neurons and regions that are critical for integrating and associating information and quality controlling what we are seeing and what we are planning and all sorts. So when psychedelics bind to these and cause all sorts of disruption, stuff goes wrong. Now, we said that psychedelics decrease overall brain activity. Well, they are highly entropic. And alongside this increasing global connectivity, or different regions of the brain that don't normally speak to each other, communicating with each other. Thank you very much. Lovely.